Okay, so now we're going to get into the reproduction of plants, which has a very conspicuous pattern called the alternation of generations. Now, all things that go through sexual reproduction produce or go through the process of meiosis, but plants and plant-like organisms also have this kind of switch between haploid and diploid stages, and that's what we mean by alternation of generations. So, a few things just about the reproduction of plants is that they can do asexual or sexual reproduction. Now, this is not what is meant by alternation of generations. Alternation of generations is just the sexual life cycle. But in asexual reproduction, they just go through the process of mitosis. They may send out a shoot or a root or something that extends the plant and then separates from it. So this is an example of vegetative reproduction in strawberries. The sexual reproduction is in nearly all plants where they have the production of seeds through the fusion of sperm and egg using pollen in angiosperms and gymnosperms and seeds. But in non-vascular plants like ferns, they don't use seeds. They have a different life cycle, which we'll talk about in class. In order to have this sexual reproduction, you have to produce gametes. You have to produce sperm and you have to produce eggs. And they may be in different structures such as zoospores or pollen or the structures we talked about within the flower. All those are important for these sexual reproductive life cycles. An egg and a sperm then are haploids, and they unite to form this zygote, which is diploid. Alright, so cells have two sets of chromosomes, one from each parent, and during the S phase of these one from the mother, one from the father, are homologous chromosomes. They code for the same thing, and they may have a different number of them. So humans have 23 pairs of chromosomes, 46 total, 23 from the mom, 23 from the dad. Plants have varying amounts and numbers of chromosomes, but they all have one from each parent. So what happens in meiosis is you start with one cell. It doubles in DNA, goes through 2 rounds of cell division, and as it does that, it splits the chromosome into 4 haploid cells, which are all unique due to events such as crossing over, where you have bits of the mother and father DNA switching between the chromosomes, and also independent assortment, where you have a mix of different types of chromosomes. You may have chromosome 1, 2, 5, and 7 from the mother, and 3, 4, 5, and 6 from the father. Okay, and this allows for each gamete to be genetically unique. So the DNA, before it splits, doubles, and it does this in the S phase of interphase to form these tetrads, and this is what people usually draw when they talk about chromosomes, which is one chromosome, but it has 2 copies of each of the genes. So these chromosomes are composed of 2 chromatids. These are sister chromatids because they are the same exact copies. During meiosis, during division 1, the number of chromosomes is reduced to half. And so each of these tetrads are separated during meiosis 1, and then during meiosis 2, the tetrad is split in half. So these chromatids, these sister chromatids, are separated. Now, in between there, though, you get crossing over between these different tetrads. Tips of chromatids will go to other tips, and that will recombine the DNA. So the 2 main stages are separated into meiosis 1 and meiosis 2. So here's meiosis 1, prophase, metaphase, anaphase, and telophase. These are very similar to mitosis, with some differences in how the chromosomes line up, and we'll talk about those in class briefly. The metaphase 2, you then end up at the end with 4 total haploid cells from one cell in the beginning. So in alternation of generations, you have a haploid cell with one set of chromosomes, which comes together as gametes to form the diploid cell, which is the zygote. There are some events in plants, especially, where the chromosomes don't separate properly. And they will create diploid sperm and egg cells, and then they will create polyploid zygotes. Okay, so this occurs if you have one cell that doubles and then doesn't split. It may have B2N, and it could combine with a 1N to form a triploid, or you could have two 2Ns coming together to make a tetraploid. And these are uncommon events, but they are common enough to where they can create new species from different formations of these genes. So triploid generally does not allow for meiosis to occur in the next generation. So if you have a triploid plant, such as in navel oranges or seedless watermelons, they don't produce viable seeds, which is good for us because then those fruits are easier to eat. A hexaploid is a six sets of chromosomes. So if you have a triploid that then doubles, it could, as a hexaploid, go through meiosis to make triploid gametes. And bread wheat is an example of a hexaploid. Alright, so the alternation of generations then is a life cycle where you have alternating between this haploid form, which is the gametophyte, and the diploid form, which is the sporophyte. So as a haploid, it can go through mitosis to form multicellular structures, and the same thing as a diploid gametophyte. Sporophytes develop from zygotes. Okay, so here you have a zygote forming this sporophyte here, which then produces spores, sporocytes, which go through meiosis to produce spores. On the other side, gametophytes develop from spores that come together through the process of fertilization and produce a zygote. Right? And fertilization then is the fusion of two gametes. So the first cell of the gametophyte generation is a spore, and then the last is a gamete. So here we have the diploid stage here. You have the formation of these spores or haploids, and then that ends with fertilization, and vice versa on the other end. Okay, the first cell of a sporophyte generation is a zygote, and the last cell is a sporocyte. Sporocyte cells contain twice as many chromosomes as the gametophyte generation. So from sporophyte to gametophyte, that occurs as a result of meiosis, and from gametophyte to sporophyte, that occurs as a result